Well, I got to click, got it. Okay. Well, thank you for the invitation. I'm happy for this opportunity to chat with all of you. I have to tell you, when I saw the email and I saw the words Chilloquium, I was so close to just hitting delete. It just sounded like some kind of spam email. So you might want to, when those invitation emails, when you send them out, you might want to bury that title a little further down in the email, do something different to catch their attention. Anyways, fortunately, I didn't hit delete. Actually, I actually read a little further. So I guess my instructions here are to talk to you for about roughly the first half an hour about my career trajectory and also what my research interests are. So I'll try to do that and keep that to just 30 minutes. I could talk for way too long just about that. So let's see. So I started I was an undergraduate at Harvard. I was a physics major. I came to Harvard wanting to study physics. And after 4 years of that, I stayed on at Harvard for graduate school. And I stayed on to do high energy theory. So this was in, I guess I started in the fall of 1985, my graduate school, and stayed there through, graduated in the summer of 1990. And that was a time when high energy theory, well, like today, it meant a bunch of things. You could be doing anything from particle phenomenology to string theory. And so although I studied, of course, the full range, I ended up doing research that was, I call it string theory. It was really what's called conformal field theory. Most of it's somewhat tangential to at least most of string theory at that time. But after, so I don't know if I should talk about that any more than that. I was studying, so conformal field theory are basically classes of two-dimensional field theories, quantum field theories, and I was in particular extending them to complicated two-dimensional surfaces beyond just the plane. I was studying some aspects of that. So after that time in graduate school, I did my postdoc, my first postdoc at UC Santa Barbara. I joined the high energy theory group there. And I can't remember now if it was initially a 2 or 3 year appointment, but I ended up spending 3 years there. And so the first year I basically continued. This was I don't know how many of you know UCSB. It's certainly it's a wonderful place to do physics. They also have one among the many things that's wonderful about it. They have this Institute for Theoretical Physics, which was running at that time and is still running. And so I interacted a lot there with people there as well as in the physics department. But basically within my first year, although I was doing well by some measure, I think, in terms of the kind of work I was doing, I kind of had a crisis and just decided I didn't want to be doing high energy theory anymore. And so that crisis, there was a 24-hour period where I thought I was going to go to medical school, or at least thought I was going to try to go to medical school. But that was just a 24-hour period, kind of got purged out of my system, partly by having a careful conversation with my father, who is an MD. So he kind of somehow asked the right questions and made me realize that I didn't really want to do that. And then there was kind of this key conversation with a friend of mine, Bruce Shaw, who was also a postdoc at Santa Barbara at the time. And I remember it vividly. We were sitting on this bench together, and I was sort of, what am I going to do with my life? And he sort of said kind of the obvious thing. He said, well, what do you have to lose? Just try something else in physics if there's something else you like. Is there anything else you might like doing? And, I already knew the answer to that, which was that I always thought, I was kind of jealous of my friends who were doing condensed matter physics, because I felt like they were doing science. They were doing what at first attracted me to science, which was trying to understand the world around me. And I had worked myself into into doing high energy theory, where the only data that I was dealing with was maybe that we live in 4 dimensions, but beyond, or at least manifestly in 4 dimensions. I wasn't trying to explain anything more concrete than that. And I think I personally was attracted to the subject for what, in retrospect, I think were somewhat immature reasons. I just, it seemed like, oh, it's very difficult and it all sounds very profound. And while the big questions in high energy theory were fascinating to me and are still fascinating, the kind of the more micro scale work, with what you're doing on a daily basis, I think it just didn't interest me as much. I found that I was really doing research because other people said it was interesting, not because I found it interesting. So with that push from my friend Bruce, I took advantage of the wonderful structure of high energy theory groups, where you can basically do what you want. At least that's how it was at Santa Barbara. And so I was able to continue working I just started working in condensed matter theory. And in fact, I did what's typical of all my friends who have changed fields in physics or dabbled in other fields. We're all very timid, and we usually try to grab onto what we already know and see if we can apply it to this new field. So, I was thinking about condensed matter theory, still not experiment. What do I know? And well, I know about random surfaces, because that's kind of at the basis of string theory. And of quantization of strings is about quantizing random, summing over fluctuating world sheets, the paths that strings take. So it's basically you're thinking about fluctuating surfaces. So I started thinking about, well, where else can I find fluctuating surfaces? And digging around, I came across, in fact, there's lots of work on that in condensed matter physics, and in fact, one of the classical manifestations of a random surface is the fluctuations of the membrane of a cell, or synthetic versions of those membranes. So the fluctuations in this case are not quantum fluctuations; they're thermal fluctuations. And the membrane usually it's a bilayer, 2 layers of molecules that are both hydrophobic and hydrophilic and self-assemble into this plane, which is fluid within the plane of the bilayer, but it's elastic in the 3rd dimension. And it's under many conditions, thermal fluctuations can be very strong. So that turned out to be fertile ground for doing theory. Many others had already been working in that area. And I managed to kind of carve out my own little area by thinking about what you might call, what I called impurities in the membrane, but the examples I was thinking of were proteins, which aren't really impurities. They have at least in a biological cell, they're supposed to be there, but compared to a pristine homogeneous membrane, I was thinking of them as impurities. So I spent my 2nd and my 3rd year at UCSB doing that, and I discovered that UCSB had an incredibly strong area. This is what I think many of you know probably, or you've heard of what's called soft condensed matter physics. And this is part of soft condensed matter physics. And I discovered Santa Barbara actually has a really strong program in that, between physics, materials, and engineering programs there. And so I got to know those people. And when my postdoc ended, they all said, oh, well, you're now doing soft condensed matter physics, so you have to do another postdoc. You have to go to Exxon. And so Exxon is in the oil company. So Exxon, Exxon, of course, is enormous, but they have a research lab that's out in Annandale, New Jersey. And at that time, at least, it was a powerhouse in soft condensed matter physics. And all the people, if you know people in soft condensed matter physics, there's a very good chance, if they're of a certain age, my age or older, that they spent time or passed through Exxon. So like Bell Labs and like IBM and many other basic research labs, much of it collapsed right after I left, right around that time. A lot of people left, and the research lab is still there. I don't really know what goes on in terms of how much basic research is done. So I spent 2 years there in New Jersey, and doing soft condensed matter physics. But when I was at Santa Barbara, I was sort of thinking about, I was thinking of what I thought were cell membranes, a theoretical physicist vision of a cell membrane. So it was sort of bio-related, and it had gotten me interested in biology. And then I went to Exxon for 2 years, and I worked on, what were sort of interesting problems to me, like a polymer that's confined within a spherical shell, a fluctuating spherical shell, like a cell membrane, liquid crystal, lamellar phases of liquid crystals under shear flow. But it just didn't really grab me somehow. And I kind of figured, this is not really what I want to be doing. So and I think what I really missed was the bio side. So that was 2 years at Exxon. So 3 years at Santa Barbara, 2 years at Exxon, that's 5 years of postdoc. And this is in the 90s when, this was now early to middle 90s when it was very hard to get jobs with PhDs. Many of my friends had gone to Wall Street and basically created all the problems that we have today in the world of finance. We'll forever have because of them. Some other people I knew went out and started working on this new thing called the World Wide Web. Well, that was more like in 1990, but they needed web designers. So there was a lot of not pressure, but there's definitely a lot of opportunity. And I considered some possibilities, what to do with my life. But right in 1995, or maybe it was 94, when I was, getting ready to figure out what I was going to do when I left Exxon, there was this ad for a new center for studies in physics and biology at Rockefeller University in Manhattan. And it had a list of requirements that were so selective that there was essentially no one in the world except me and possibly one or two other people who met their requirements. And I don't mean requirements as in great achievements. I just mean it just had very restrictive requirements. So they wanted you, first and foremost, they had some very polite way of saying you had to be kind of old and had not yet gotten a job. And I forget how they actually phrased it, but mature or something like that. You had to have been a theorist. You have to be a theorist, and you had to have done at least a little bit at the interface with biology already. And back then, there really weren't that many people who were theoretical physicists who were dabbling in biology. So with all those requirements, I think I applied. There was already to this position, it was a fellow position. There was already one fellow, the first fellow in this program, that was John Marco, who has gone on to a great career. He's a professor at Northwestern. And so I got that position. And basically the position was, you come to Rockefeller University, you have 3 years and you can do what you want. So many of you in physics may not know about Rockefeller University. It's a very odd place. It's a tiny little oasis on the Upper East Side of New York. It's right on the East River. It has an old history of, and a tradition of actually doing a lot of great breakthroughs in biology. Bizarrely, at least at that time, they did have a high energy physics group there, a very small one, which I don't remember why, but there's some history behind that. And then they had recruited Mitch Feigenbaum and Albert Liebscher at that time to really kind of start this center. And they started getting fellows, and they hired some other junior faculty. So I thought I was going there. I thought this was basically career suicide, right? Because 5 years of a postdoc, and now basically switching fields where I'm going to do biology or something at the interface of biology. And this was 1995, where at that time wasn't clear if there were going to be any job opportunities for something like that. So the only real reason I went there was because I love New York City. And I figured it would give me a way to stall and figure out what to do with my life. So I could go to New York and hang out, and they'll pay me for 3 years, even if I accomplish nothing. And the reason I going out of my way to say that is because I actually think my coming there with that attitude was kind of critical for me to really end up landing on my feet, because I really was no longer approaching my kind of post PhD period with eyes towards getting a faculty job, doing something that's going to somehow impress somebody else or somebody else might find interesting. I think I was there, I was still showing up every day, but I was just going to kind of have fun because I really didn't feel like I had any chance of getting a faculty job at that point. And I think that was what enabled me to really find out what I love doing. Although it was still a long and rocky path. I ended up spending 5 years there. So I spent 3 years part of this fellows program. They extended it by a year, and then I stayed on 1 additional year in a lab. So I started out by continuing exploring questions of membrane biophysics that I had really started already back in Santa Barbara. And I started very quickly. Within 6 months, I realized I don't want to be a theorist. I want to do experiment. And I think I had always really liked doing experiment. And I spent a big part of my time in graduate school hanging out in my friend Charlie Marcus's lab as he was doing experiments, and I would just sort of hang out and watch him, which probably I should have paid attention to at that time. Was that I was attracted to doing things with my hands. So again, that's an incredibly reckless thing to be doing this late in your career, you could argue, but I felt like I had nothing to lose. So I started doing some problems in membrane biophysics, measuring electrical signals through ion channels and looking at the effects of surface tension. And it was sort of around that time that I started reading about electrobiology and really experimental aspects of microbiology. And that was when everything just kind of clicked for me. And I really found out, this is where suddenly I really found what I loved. And I think what happened is it was some kind of combination that I've always had a love for biology, even though I'd never taken biology since high school. I think I'd always found it interesting. But I also loved electronics. I had taken, there's a course, I don't know if it's still called this, at Harvard called Physics 123. And I took that course from the person who taught it, Paul Horowitz, who wrote the book, The Art of Electronics. And I just loved the course. I loved learning it from him. And so it was kind of a closet love of mine. And reading about microbiology, it just felt like electronics, only with squishy components rather than transistors and resistors. And you're integrated circuits, you're dealing with these soft materials for which there's still information transfer. You can cobble them together in principle to do new things, or you can try and disentangle them to figure out how they work. And so I wandered through a bunch of different projects. Most of my time at Rockefeller, I failed miserably. The problem, the labs, there are many labs that are very welcoming. I was free. You could walk in, sure, you can come and work with us. And I spent most of my time in the laboratory of Sandy Simon, who broadly is interested in almost anything any process that involves crossing a membrane. At least that's how I would describe it. So he had this wonderfully broad interest, and he had a great group of people in his lab. But I think part of the problem is that I was so senior based on my number of years out of graduate school. I think people in some sense assumed I knew more and had more experience than I really did. I knew less than the first year graduate students that were in his lab in terms of molecular biology. So I think I was given a little too much rope at times, but I definitely learned how not to do a lot of things the hard way, how not to do a lot of experiments, but somehow enough worked that I was somehow managed to survive. And I spent most of my time there actually working on studied aspects of animal cell biology, all of it with a very physicsy bent, I guess. Again, going after mechanisms of how the nucleus of the cell controls what does and doesn't get inside the nucleus at a physical level. I also spent time trying to understand how certain viruses self-assemble and get out of the cell. Most of those things failed just because I had a wildly unrealistic idea about how to go about the questions. But during that time, I really fell in love with microbiology and bacteria. And I really trace all of that to a critical talk, a seminar. I was running the seminar series, and someone had recommended I invite Tom Solhavi, who's a microbiologist at Princeton. Someone recommended I invite him. And he talked about E. coli and the response to osmotic pressure. So you remember osmotic pressure is basically associated with how much solute is dissolved in solution. And in particular, there's a osmotic pressure difference across the between the inside and the outside of the cell. He was talking about a system that can detect that difference. And then the cell responds and does these mysterious things where it changes properties of the envelope of the cell in response to this osmotic pressure difference. So I immediately got really interested in the question, well, how do you detect an osmotic pressure? And you might guess that it might be membrane tension, which is one possibility. And how can you go about testing that? Are there other ways a protein might be sensitive to the osmotic pressure difference across the cell? So again, it was really kind of a physicsy question, I guess, that got me sucked into that. And it's actually, I spent a long time studying that problem, even after I came to Penn. I no longer really am convinced it detects osmotic pressure. So it's, I've never really necessarily answered the original question that interested me. But I really fell in love with working with bacteria, and especially E. coli. And again, this partly goes back to this level of electronics, where E. coli is really it's kind of like the machine shop for doing molecular biology. It's the setting where if you're going to be re-engineering DNA sequences, it's at least usually the first place people go to try and do those kinds of manipulations. And there are so many tools available for studying E. coli. So that's where I decided I wanted to spend my time. So when my time ended at Rockefeller, I was applying for jobs. Because my girlfriend at the time, who's now my wife, wanted to stay in the New York area broadly defined, I kind of limited where I was applying. And in the end, I had an offer to go to Penn in the physics department, or I could go to, I also had an offer at a biotech company, believe it or not. And at the time, it wasn't obvious at that point which one I wanted to go to. Both seemed kind of exciting. And in the end, I chose Penn because I think I had this sense that it would probably be easier to go to biotech from an academic position than vice versa. I don't know if that's necessarily true, but I think that was really the reason I decided to go the academic route. But I'm happy I did. I haven't gone to biotech. So I stayed in the physics department. And then at one point, around 2006, I think it was, I took a joint appointment. So now I'm in both the biology and the physics department. And my lab is now in the biology department because it just kind of made sense because I really kind of went so far into the biology side of the physics biology spectrum that it made more sense for me to move my lab over here where I am now in biology. But I still have my office in physics, and I still do some teaching in physics in addition to biology. I've already talked for half an hour. So as far as what I do, well, Andrew kind of really summed it up pretty well. Very broadly, I'm just fascinated by what I think of are the circuits in cells, and especially the circuits in bacteria. And by circuits, what I mean are the mechanisms that bacteria use to sense what we might call signals or cues in the environment. They process that information. You can think of it as doing a computation almost, right? They're taking in information, and then the cell will then there'll be some kind of response based on that information. And it's something, those of you who study biology sort of in some ways take that for granted because you're just inundated with information in biology. But if you sort of step back or you think about it from the perspective of a physicist, maybe, or maybe an engineer, you could say, these are, on the one hand, these sound familiar to us like the engineered devices that we have around us, but they're not built out of silicon. They're built, again, out of these squishy materials, right? They're built out of proteins, carbohydrates, lipids, nucleic acids. They're subject to significant thermal fluctuations, and the flow of information is not governed by electron flow through wires. Sometimes it is charge transport in the context of biology, but it's still not. There's no conduit it's going through. So whatever the flow of information is, it's again, it's going to be sloppy in the sense of it's governed by statistical mechanics. And in fact, much of it is due to molecular interactions. So this question of what are the governing principles that, what are the principles that govern what kinds of these squishy circuits, what they can and can't do, and more generally, how they evolved, because these are evolved circuits, these aren't designed circuits. And so what are the constraints imposed on them just by the fact that they have evolved are really fascinating and open questions. I don't really have illusions that there's going to be a clear-cut, tight answer to this, but it's fascinating to try and explore these questions in the context of different regulatory systems. And I'm not the only one, of course, who's asking these questions. Many people are motivated by these questions. Not everyone says it the same way I do, but they really are asking the same questions. And you can ask this on everything from neurons in the brain to fungi, plants, or bacteria, which is where I chose to focus. I better stop talking, so you guys, in case you guys have questions.
Yeah, as before, you can raise your hand and ask the question, or you can send them to me and I'll ask them. And in the absence of questions right now, maybe take a few minutes. We have some RSVP questions as well from our RSVP form, which I can go ahead. Okay, I'll go ahead and ask those first. So the first question is, how can this research be related to recent studies that seek to use bacteriophages alongside antibiotics to combat resistant bacteria or superbugs?
Well, so my own research is very much at the heart of both combating antibiotic resistance directly in order to allow antibiotics to be functional, as well as the use of bacteriophages. Those of you who don't know about bacteriophages, those are viruses that attack bacteria. And so I've been very interested in those questions, not necessarily all of antibiotic resistance, but a subset of those. Many of these what I called circuits that we study, many of them actually are involved with one form or another of antibiotic resistance. So for example, the one that I first mentioned to you that got me interested in the subject, that was alleged to be a system that responds to osmotic pressure, what it actually ends up, the output of the system, never mind what the input is, because I think that's still an open question, but what the output is among the many outputs, one of them is the permeability of the envelope of the cell. And so and that changing that permeability affects how readily antibiotics can penetrate into the cell. And so when the envelope is made less permeable, when the cell either mutates or in response to an environmental signal lowers the permeability, then intrinsic pumps that the bacterium makes are able to pump the antibiotics out fast enough so that the cell can survive at least some level of antibiotic. When the permeability in is too high, then the pumps can't keep up. So changing the envelope permeability is in fact one of the very common mechanisms that naturally acquired antibiotic resistance, at least certain levels of antibiotic resistance, that you'll find that in the clinic, for example. Bacteriophage, so phage therapy, if you read, there's a famous book by Sinclair Lewis called Aerosmith. So if you're interested in phage therapy, you can go back to that book. It's fiction, but it's kind of based on the early days when people had discovered bacteriophage and thought about the possibility of using these viruses that target bacteria as a way to kill off bacterial infections. And so that has come back. That it lost popularity because people realize that bacteria get resistant really quickly to these viruses, but it's come back because people have come up with a number of different ways to try and counteract the emergence of that resistance. So we got interested in recently in some of these bacteriophages because in the context of phage therapy, because we were studying an antibiotic resistance mechanism where it's an enzyme that many bacteria can pick up They can pick up a piece of DNA from the environment that carries a gene which modifies the charge of the cell envelope. And so it makes the cell envelope less negatively charged. The cell envelopes of most bacteria has a net negative charge. And many antibiotics are positively charged. And by making their envelope less negatively charged, they can become much more resistant to certain classes of these positively charged antibiotics. So we were studying one of these resistance mechanisms, and we figured there's got to be a fitness cost for the bacterium to pick up that particular gene. And we tried a bunch of things, and we discovered that this was in fact made the bacterium more sensitive to certain bacteriophage. So we've been sort of thinking about the possibility then of using that as a mechanism for purging this form of resistance from a population of cells. There's definitely a question that resonates with me.
Yeah, and actually, I have a question that's been on my mind a little bit with respect to the interaction between biology and physics. Are there many or any biological processes wherein, say, like quantum mechanical effects have like physical significance, or biological, maybe I'll say biological significance? Like if we were to say simulate some biological system like a cell, what kind of tolerance might we see if we remove any quantum mechanical effects, if we just try to do this classically?
Yeah, so of course, there are many things that, to the extent that there's certainly a lot of chemistry underlying a lot of these processes. So the boring answer, which I don't think you're thinking, I know you're not really thinking of this, but I should at least say the boring answer first, which is that, what's governing a lot of these chemical reactions are, in many cases, quantum mechanical effects. And in many cases, you do need to use some quantum mechanical simulations of some kind if you want to understand some aspects of some of these chemical reactions that go on, just as you do in all kinds of organic chemistry or any kind of chemistry, I guess. But what you're, I think, asking is more on the spirit of something that's kind of on a slightly larger scale, so to speak. And so the one example I know of has to do with the electron transport chains. And so electron transport chains they govern, first of all, the process of respiration, energy generation by respiration. So you have molecules that are electron donors, and they're in some sense in a high energy state. And what happens is they can donate electrons into a pathway that lives in the membrane of the cell. Well, in bacteria, it's in the membrane of the cell it's in the membrane that bounds the cell. In humans and other animals, this is all going on in a little membrane-bound organelle called the mitochondrion. But in any case, the idea here is the electrons get donated. They enter this electron transport chain, and then the electrons are moved. They're passed along. And as they're passed along from one step to another, they're literally getting passed from one protein or other type of molecule. There's some other molecules that are carriers. Each time, a little bit of energy is given up, and then that energy is used to create an electrochemical gradient of protons in most cases across the membrane. So it's basically like charging a battery. And the fact that it's passing the electron is losing energy on small steps is something you may have already been familiar from your thermodynamics courses, where that's kind of a general, I don't think it's really a principle that I know of, but it's in practice, if you want to get as much efficient energy out as you can to use for your useful work, you want to actually remove the energy in relatively small steps, usually, rather than just trying to do it all in one big step, like an explosion, where you're going to lose most of it as heat. So, so to go back to your question, the question is, a lot of those some of those electron transfer steps are basically classical physics, but it turns out that a number of them look like they are quantum tunnel, and they really are tunneling from one end of the protein, for example, to the other end. There are these cofactor molecules that are carefully positioned within the protein, and as I understand it, I'm by no means an expert in this field, but as I understand it, in order to understand the charge transport through there, they do have to model it as an actual quantum mechanical tunneling process. So that's one example. Photosynthesis, I guess, is the same. That's sort of roughly the reverse process where you're using light in this case to charge batteries, but and make oxygen. But that also is, I believe, has a quantum mechanical component. Those are the only ones I know that are mainstream. Other ones are, there are some unusual theories you'll find out there, people claiming quantum mechanical effects of to try and understand consciousness. I think Roger Penrose may have written some papers on that subject, but I don't think those have gained very much traction as far as I know.
Right, and a question in the chat now. So that is two of them here. Which skills would you say people should learn if they want to work at the intersection of biology and physics? The second is, second is, which courses, apart from specific biophysics courses, would you advise taking? For example, electromagnetism, cell biology, and so on.
Yeah, I get that question a lot. It's a good question. You know, so oddly, I sort of feel like what's first of all, what's most important is I would go after the courses that interest you. It's really hard to explain, but you know, if you take a course in you know, I don't know, quantum mechanics, or if you take an advanced course in electricity magnetism, which by and large is not probably going to be that useful for what you're doing in biology itself. I feel like what's really going on, it's not so much about the actual things you're learning as much as sort of how to think and how to solve problems. And especially, I think well, I can only speak for a physics education because that's what I had, but I think there's an enormous amount you learn that they're almost like, it's not that you're directly applying that information. It's you're learning way more than you realize when you're taking those courses. So first and foremost, I would say is probably any sequence that you take, in my opinion. I think physics, for example, is a great preparation for going on into biology. Even if you take relatively few biology courses, I would err on the side of more physics courses than biology courses. I can say with experience, there are some, I can pull up my bookshelf here, I can pick out some textbook physics books and point to some pages where I can tell you, I will never understand what they're trying to say on this page. Not because it's badly written, but because of, I can't hold everything in my head to fully understand what they're saying. My experience with biology is it's less like that. And it's somehow, the challenge in biology is there's a huge amount of information. And there are definitely some deep ideas, of course, but somehow assimilating that I find is not as difficult. And so if you end up mostly studying physics and then you pick up the biology, say in grad school, you'll be able to do it. Whereas the reverse would be a lot harder, I think. If you studied only biology as an undergraduate and you went to graduate school in physics, I think it would be something about the nature of the subject and how it builds on itself so much. There's so much of it, the assumption about the basic classes you had in physics and what you know and how you think in physics. You would find you'd have a very difficult time making that transition. So that's my first advice. But to go back to your question, I think statistical mechanics is really important. So I would definitely say take a lot of statistical mechanics. Take more than one course if you can. It really is at the heart, I think, of understanding so much of what's going on in biology. Definitely a good biophysics course. If you can take a soft matter physics course, if your school has one, that's nice because it's applying a lot of statistical mechanics, but in a context you'll be exposed also to sort of the zoology of soft matter physics, so much of which is relevant again in the context of biology. So those are the ones I would say to me are most important. And then you know, if you have an opportunity to learn a little bit of like a fluids course, again, that the tools could end up being very useful for you, but but it comes up a lot, surprisingly. A good fluids or even better complex fluids course. So if you can learn about Smolakowski equation and diffusion equation, that could come up in an advanced stat mech course too. Just kind of depends what you have access to. But I guess the key point is, what's going on in the cell is definitely, it's mostly thermally driven. And so that's the realm, to the extent you want to draw on physics ideas, that's where you can take it. The other thing, of course, is chemistry. Chemistry is important too. In my experience, physicists have a kind of fear of chemistry, and they try to avoid it. But if you can pick up some chemistry, it'll make your life a little easier too.
And sorry, as for skills, so like I was thinking coding in some language, especially R and Python, but I don't know if there's anything else that could be very useful.
Yeah, so coding, so that's definitely, it's not a requirement. It depends on what kind of biology or biophysics course you go into. But my attitude with coding has always been, it's less about learning a particular language, is more about just kind of knowing how to code. And sort of once you learn one, you usually can pretty much quickly pick up something else. But R and Python definitely are the two by far that dominate what people are when they're trying to process lots of information that's out there. So those are really the ones that I would say. Beyond that, if you're into it, things like, I guess, a course on algorithms or something like that. Definitely people here that go into computational biology, for example, they're often taking a good course in algorithms just to learn a little more about what's under the hood.
And a question from the audience, and Minchell, you can go ahead, ask your question.
Yeah, thank you so much. Yeah, so I think the systems we deal in physics is much more like abstract compared to the system in like biology. So I just wonder whether the training in physics is kind of like helpful for dealing with those kind of like complexity or like non-abstractness of the biological system, and then like reduce those kind of system into the more abstract version of that. And then if so, how those kind of things can be related.
So I guess there are different ways of interpreting what you're calling abstract. I wonder if you're really thinking in terms of maybe simple systems, if that is maybe what you mean. I mean, I remember when, there are certainly physicists can work on what on the face of it look like very complex problems, but I think maybe what you're thinking of is they tend to try to distill it to a very simple form. Is that what you mean, right? Right. So like if you're a physicist studying turbulence, you're probably less likely to go out into the open ocean during a storm and study that, right? You're more likely to build the simplest system that makes turbulence in the lab, right? Where you can control it, and right. And so in biology, this is definitely always a challenge for physicists. And in some sense, the fact that I've stuck with bacteria, which to you probably maybe they still sound hideously complex, and they are. By some biologists' measure, they would say, well, they're very simple. And so for me, I think maybe that's part of the attraction that I've had for studying E. coli. I think for everyone who goes into biology, there's this kind of spectrum of complexity versus, or complexity is really the wrong word, but the extent to which you have control over your system. And there really is this big spectrum. And so it's a matter of taste. There are the people who will, literally try to model they want to study protein folding, and they'll model it as an Ising model, right? And, people, they made some progress early on. Usually people kind of get their feet wet by doing that, and then very quickly realize, well, I have to kind of go beyond that to make further progress. But so often that sort of physics tradition of really the spherical cow, or whatever you want to think of, that a lot of people who first take a step into biology, that's what they do. It's again kind of the safe place. But then the more they're driven towards trying to find kind of a relevant the biological relevance, they need to start adding complexity. But that said, there really is an art within biology, and I can't say I've mastered this, but many people have, of taking a complex system and still just kind of realizing you can focus on a small subset of easily defined variable, easily measured variables that you can put under control, and you can actually explain what seemed like a very complex phenomenon. And there are a lot of examples out there. They're usually people with a physics background who are doing it because they're sort of driven to do that because of their physics background. So that, but I guess it's a little different than what you're used to. Usually in physics, often you can neglect, you do that because you choose a length scale. So you're going to describe what's going on at this length scale, and you sort of average over everything at a smaller length scale, and you sort of automatically get a simpler model. But in reality, there are definitely examples within physics where, in fact, it was a much more intuitive and at least in condensed matter physics, there are examples where it was people came up with models that weren't necessarily. They kind of made some really crude approximations, and they threw out a whole bunch of things. So let's just assume we can throw all this other stuff out. And sure enough, they were able to describe the essence of a complicated phenomenon. And so that you can still do in biology.
All right, another question from the audience. Heather, you can go ahead and ask.
Hello. So I'm a biophysics major, but I'm on the track towards medical school. So I'm, as someone who has like dabbled with the idea of going to medical school, I'm curious what advice you might have for someone who's interested in both of those fields.
Biophysics, you're saying biophysics, who's interested in biophysics and medicine or medical school, that's what you're saying. Okay. So first of all, I can tell you that I think medical schools love physics majors. I don't know if it's because most of the people who are interviewing are terrified of physics or something like that, but I think they actually they appreciate that you've had a really rigorous training, and so there's that. I know that's not necessarily what you were really asking, but there's huge opportunity there. I mean, one of my friends back when I was in Santa Barbara, he was doing low temperature physics, and he took the bare minimum number of courses and applied to med school, and you know, they took them because so you don't have to worry on that front. But it's, you know, in terms of opportunities, they're enormous. I mean, there are many people. So I guess I don't know if you're thinking, are you thinking of an MD PhD possibly, or straight MD, but maybe you still want to do some biophysics?
The second option, I'm thinking.
The second option, okay.
Mm-hmm.
So, you know, so the one obvious overlap are things like in radiology, for example, where there's a lot of physics, and a lot of those people, either they have kind of a physics background already, or they need to pick it up, because so much of the tools in radiology, the different kinds of imaging systems and contrast approaches to contrast are based on physics. But you know, that's sort of the obvious one. But the reality is, many other areas within medicine involve aspects of biophysics, you know, physiology, all of the homeostasis in the body. If you haven't already, when in medical school, you'll learn, right, about all the different control systems that enable us to maintain our temperature at 37 degrees, and all of that will resonate with you as with your training and your background on biophysics, and you may in fact be drawn into research questions that in fact involve biophysics. So it's hard to point to any one single opportunity. I should also mention to you though that even as with straight MD route, you can still end up doing laboratory research. And in fact, there are many examples of people who got an MD, they did a fellowship afterwards where they started working in a lab, and then realized, you know, I don't really want to see patients. So they just end up being full-time, basically as if they'd gotten a PhD in some cases, or maybe they go into the clinic just occasionally. So that is always an option for you as well, if you go the straight MD route and then decide you are particularly excited about some kind of some particular area in biophysics.
Thank you.
And another question in the chat. Could you please ask Dr. Goulian what he meant when he said bacteria swallow up genes? where exactly are these coming from, and is the endocytosis of the genes also related to the bacterial cell permeability?
So I don't remember if I actually used the word swallow up, but I like that way of describing it. So this process of DNA getting exchanged between bacteria is what's called lateral gene transfer or horizontal gene transfer. And there are actually a bunch of different ways that can happen depending on the bacterium. So there are certain classes of bacteria that are what we call naturally competent. And the competent there, it means they're competent for taking up naked DNA from the environment. So the most famous example is Streptococcus pneumoniae, which back in 1915, I think it was, where there were these famous experiments where they didn't know what was going on. This was sort of at the beginning of people really understanding that DNA was a genetic material. And so they were able to see that they could transform a bacterium into a different bacterium by dumping the guts of dead bacteria on top of them. What they were doing was dumping the DNA of those dead bacteria. It was getting taken up in these other live cells and incorporated. So that's one mechanism. How that happens is fascinating. It's there's a whole machinery that does it. There's some kind of import machinery that brings the DNA in, and then it through a process called recombination, it kind of pasted into the chromosome. Another mechanism, the one I was actually thinking of, is where it's actually coming from another bacterium, and it's a little bit like mating. It's literally in fact, we use the word mating. One bacterium will grab onto the other bacterium, and through a pilus, this long structure, the structure shrinks, brings them close together, and then through a process that incredibly we still don't understand, even though this was discovered in something like 1950. Through some process, the DNA is transferred from some DNA is transferred from the donor bacterium to the recipient bacterium. And in this case, this is actually one of the major modes of transfer of antibiotic resistance genes, in fact, between bacteria. So the other bacterium then picks up this piece of DNA, which usually circularizes into a small little mini piece of DNA we call a plasmid. But then will replicate and stay with that bacterium, but then it can go on and donate it to someone else. So that's another mechanism. Another one is viruses. So viruses can sometimes, instead of packaging their own genome, which is normally what a virus does before it breaks out of a cell, sometimes they accidentally package some of the bacterium's genome instead, kind of by accident. So you end up getting this viral particle, but it no longer contains a viral genome. It contains a piece of the bacterial genome, but it's still able to infect another cell, another bacterium. But when it infects that bacterium, rather than injecting a viral genome, which would then create more virus particles, it injects a piece of bacterial DNA, which with some low probability can then integrate and get pasted into the genome of the recipient bacterium, and suddenly that recipient bacterium has gained a bunch of new genes. So those are really the sort of roughly the three major methods by which genes can be passed between bacteria. So it's not really endocytosis, like you mentioned. Bacteria don't really do endocytosis like animal cells do. They have a fairly rigid cell wall that, among other things, would make that kind of challenging to do. But they definitely can exchange DNA, and in fact, they do it at much higher rates than other types of cells, which makes it a fascinating subject. I've really been fascinated by lateral gene transfer because I like to think of it as it's as if you had like an electronic device, and there's sort of this constant flux of random electrical components entering the computer and randomly inserting themselves into the circuit, and with some probability they actually provide a new function for the circuit. It sounds like a crazy way to produce a new electrical device, but on the scale of biological cells, it's an incredibly powerful way to evolve new functions.
We have time for maybe a couple more questions. Shreya, you can go ahead and ask your question.
Hello. So I'm kind of curious about like the intersection between physics and neuroscience. I know there's a lot of progress being made in all these novel imaging techniques, but I'm kind of curious, have there been like any concrete physical models in the self-assembly of neuronal circuits? Like what does that look like?
So this is definitely at the edge of my understanding. I will tell you that I left this out of my history. When I went to New York City to go to Rockefeller, I was on the edge of going to UCSF. They had started a program where they were trying to suck physicists into neuroscience, and I almost went there, but I rather wanted to go to New York City. So definitely, first of all, there's a huge tradition of physicists going into neuroscience. I don't know if you know, like the whole theory of neuronal conduction, the Hodgkin-Huxley, or action potentials they're called, is due to Hodgkin and Huxley from in the 50s. They were physicists, really. And it's a beautiful subject if you've never read about it. And it will warm your heart because it's just a beautiful example of physics. But so at the individual neuron level, how a signal propagates through the neuron is already biophysics. You're asking at sort of the higher level, I think, now how neurons assemble. And I don't know the subject well enough. What I can tell you is it's incredibly active and full of physicists, and it's been incredibly successful. And it's an area where I think the contributions of physicists are really highly appreciated. You've probably heard of the word neural network, which goes back to John Hopfield, I guess. I think it's John Hopfield, but which I guess has become a big thing in machine learning. But as I understand it, his real interest was in describing neurons and neural networks, real neural networks. And I think aspects of those models, of course, they've been refined and they're much more sophisticated than his original models, but they all go back to basic ideas, again, in statistical mechanics, spin glasses, and all of these kinds of things that, at least over the years, just by osmosis, I heard people were playing with these ideas in the context of neuroscience. Imaging, of course, is a whole nother area. Definitely, it's another area that can be incredibly physicsy, right? And different ways of actually imaging neuronal activity. I don't know how much super resolution imaging becomes important in neuroscience, but there's all of that area as well when you're trying to push the resolution limit of light, use light to measure things well below the wavelength of light. So there are in the optical physics realm, basically, there have been amazing developments in that subject as well. So it's kind of, I'm not really giving you a very precise answer other than to be encouraging and tell you it is definitely a very fertile area and it's full of physicists. You'll have no shortage of finding opportunities as a physicist in neuroscience.
Thank you.
And if there's time for one more question, we have another one in the chat.
Sure.
Okay, so question is, can I study genetic engineering, particularly CRISPR, through biophysics?
Yeah, I mean, well, I can't speak for it. I guess that depends a little bit on your own program, but whether you're a biophysics major, a biochemistry major, a biology major, or I think most programs, there's so much overlap between those fields that there's no barrier to doing that. Bioengineering is another one. They almost just become different words and they have different flavors of studying very similar things. So yeah, genetic engineering, you can easily end up doing as a biophysicist. You can, you may at some point you may stop calling yourself a biophysicist if you end up just working on genetic engineering, but certainly CRISPR technology comes up all the time for biophysicists who are trying to re-engineer cells to study so that they can study some biophysical process.
Sorry, Professor, if there's time for one more short question.
Yeah, sure.
Awesome. So the question is, what interdisciplinary fields which incorporate physics do you see growing in the next few years?
Wow, I don't know if I can speak for all of them. I mean, biology is no sign of it shrinking, in terms of the physics. There's been a long history of physics physicists going into biology with great success. And it just keeps growing. I mean, I don't know if you guys are aware, but phase transitions is just all the rage now in biology. The number of cell biologists who are talking about phase transitions, and I'm thinking to myself, I'm not sure they know what they mean by the word phase transition, but they just so happily adapt it. the people who introduced those ideas, of course, were physicists, and so these liquid phase transitions, going on within the nucleus of cells and all of this kind of thing. So my guess is it's going to be biology is where there's certainly going to be the biggest continued growth, because it's such a big field. I don't know if I can point to other areas, just because I don't know enough about geology or economics. I hope we don't keep getting physicists going in economics. We've caused enough problems. What else? What else is there? Politics. Yeah, I guess that we haven't seen that happen yet. Maybe physicists can get into politics and do something revolutionary. I doubt it.
Yeah. Okay. Thank you so much. There are a couple of more questions coming in now. I don't know if there's. Okay. Well,
I have no problem.
So we'll take this one and then we'll close. What models from soft matter physics do you think have compelling applications to bacteria or biological settings?
Oh, it's hard for me to think of a single model because, it's the usual thing in physics where you have these sort of sequences of increasing complexity. Like I mentioned the Ising model, right? Ising model, which is not unique to soft matter physics, but is certainly used in soft matter physics. And that definitely has a place in modeling biological systems. It's the first way you model an ion channel, two-state device in the membrane. If you want to start thinking about coupling between adjacent ion channels. Polymer physics comes up in so many contexts within thinking about protein folding and packing of DNA. Of course, you very quickly end up going beyond the simple idealized models for polymers that you first learn in an elementary soft matter physics course. But if you go into more advanced soft matter physics, you learn about polyelectrolytes, right? Stiff polymers and then polyelectrolytes, and all of those start becoming important. So it's hard to pick out a single model or even a single area of soft matter physics. It really kind of permeates so much of what goes on. If you want to try to model anything in the cell, you're probably drawing on some aspects of soft matter physics broadly defined, whether it's, I'm including things like diffusion, for example, as part of soft matter physics. And because these are complex fluids and complex materials ultimately that are making up these cells. And there's great precedent now for it mattering, right? So something that's a big deal here at Penn are the material properties of cancer cells versus non-cancerous cells. And, they have very different properties. And it's been a really fruitful area for people to study who come from the soft matter area.
Wonderful. So we should probably close now. For those of you with questions that didn't get a chance to answer, well, hopefully at next week's talk, you can ask them to the next speaker, or ask some questions to the next speaker. But thank you so much, Professor Goulian, for the talk
Absolutely.
today.
It was a pleasure
to have the opportunity to speak with you. And thank you to everyone here for coming this week. We have, again, talks every Monday at 4:30. I will note, though, that next week our talk is going to be at 4 p.m. because our speaker is in Europe. It'll be Professor Dr. Jonathan Holm from ETH Zurich, speaking about quantum information science and quantum computing. Okay, thank you very much to you all for coming today. And
good luck to all of you.